ChemBioChem Virtual Symposium Chemical Translational Biology. My name is Ruben Ragg and I'm the Editor-in-Chief of ChemBioChem. I am, and we from Chemist Europe are extremely excited to host this virtual symposium for you today. With me will be Anne Nijs, the Editor-in-Chief of the European Journal of Organic Chemistry, EUJOC. Anna will support us behind the scenes and try to answer general questions that come up in the public chat that you can find in the bottom right corner in your browser. You can use the chat to communicate with each other, but also to raise questions during the talks. Please start your questions to the speakers with @Yael, @Jeremy, or @Vishal to help us identify the questions more easily. Today's seminar is brought to you by Chemistry Europe, your European Chemical Society publisher, representing 16 chemical societies from 15 European countries, including over 75,000 chemists. Now, and without further ado, let's hop over to the scientific part. We are very, very grateful to the three speakers of today's session. Yael David from the Memorial Sloan Kettering Cancer Center in New York, Jeremy Baskin from Cornell University, and Vishal Rai from IISER Bhopal. Each presentation will take about 20 minutes plus roughly 5 to 10 minutes Q&A. Now, let's hand over to this first speaker of today, Yael David. Yael is originally a biologist and switched over to biochemistry during her PhD at Weizmann with Ami Navon. And finally, she entered the realm of chemistry and chemical biology during her postdoc with Tom Muir at Princeton. So she's truly a role model in chemical translational biology, so to speak. She's now a professor at the Sloan Kettering Institute in New York City, where her lab uses and develops chemical biology tools to study epigenetic events and their influence on various diseases, particularly cancer. She's won numerous awards and is by all means a rising star in the field. But more importantly, she's a wonderful human being, always a lot of fun to have around and work with, and a great mentor to her exceptionally talented team. Yael, we are extremely grateful that you are here today with us, and we are looking forward to your talk. The stream is yours.
Wow, I wasn't prepared for this introduction. And also not ready to have my slides show up like this. Wait a second, here we go. Thank you very much, Ruben, for this very kind introduction and for the opportunity to speak here today. I always knew I'm Ruben's favorite, but I will not confirm nor deny there was a coin toss regarding who speaks first. And today I'm very, very excited to tell you some of the work my lab has been doing in the past 4 years at Sloan Kettering. I'm sorry. I don't know why this is. Okay, here we go. No, I can't hide this. So I think
the slides come in nicely, Yael.
Oh, but I can't move them, so.
Okay.
Yeah. So I'm going to have this command line still stay here. Okay. So before I move to my actual science, I want to shamelessly advocate for our Chemical Translational Biology special collection, to which myself, Ruben, and Rob Osland from Merck in Boston have collected a number of really exciting research, and we're hopefully will be coming out to you in ChemBioChem in the next few months. Okay, so my lab is primarily interested in understanding fundamental mechanisms related to epigenetic regulation of transcription, and specifically how misregulation of these events can lead to disease states. And today, as Ruben alluded to, I will speak as a chemical biologist or a protein chemist and tell you about some powerful methodologies we developed to apply to address some of these convoluted questions and identify specific epigenetic events that drive changes in transcription. Now, on the methodology end of the chemical biology, today I'll mention some of the methods we have developed, specifically chemical probes to probe, manipulate, and track chemical modifications on histones. But we do have a lot of work to site-specifically manipulate histones in vitro and in vivo. So if you're interested in using inteins for some of these work, please call me afterwards. And today I'll focus on our biological question, which is understanding chemical reactions that happened on chromatin, and specifically link environmental and metabolic changes to transcriptional output in the form of non-enzymatic covalent modifications. We do have a lot of work on linker histones and non-canonical histone ubiquitination, so if you're interested in any of those, also drop me a line. Okay, so in its fundamental form, epigenetic regulation is the dynamic transition between a transcribed and a non-transcribed state for a specific genomic region. And these are facilitated by numerous mechanisms, including transcription factors, non-coding RNAs. But today I'll talk about histone modifications, which is the focus of my lab. And in this case, you have specific histone modifications that decorate the proteins, the histone proteins that spool the DNA, and change the accessibility of the DNA to allow the transcription machinery to dock or exclude it from docking. And what happens in cancer cells often is that regions that are supposed to be transcriptionally repressed are now being transcribed, and those are oncogenes, for example, and vice versa. So regions that are supposed to be tumor suppressors are now being overexpressed and drive tumorigenesis. Now, as biologists, often you think about these events as sort of these beautiful drawings, but as chemical biologists or as chemists, we know that these events do not happen in buffer or on this really nice clean page, but in fact happen in a very dense chemical environment that include metabolites, cofactors, etc. And what happens in diseases such as cancer, where metabolism is shifted, you actually have even a more diverse and more dense chemical population that can react with chromatin, chemically react with chromatin. And this is sort of our starting point from my lab, trying to understand some of these chemical reactions and how they affect chromatin, how they affect transcription, and how they drive diseases. And this has primarily been driven by the epidemiological correlation between environmental and metabolic stress and diseases such as cancer. And what I want to propose to you today is a mechanism where chromatin itself is damaged or reacts with metabolites in the environment, leading to chromatin or histone damage that can drive diseases. Now, beyond understanding these fundamental events that happen in cells and in organisms, we also try to identify new vulnerabilities in these cascades in order for us to target them. And the question we ask is, could chromatin damage actually serve as the metabolic link between environment and cancer, and can we target these? So this is already outdated, but we're working on a new review to sort of overview some of the chemical modifications that occur in proteins in red, and gray also, on histones, and gray also protein non-enzymatic modifications that haven't been identified on histones, but we're working on it. And I think you can appreciate from this table that there are various chemical reactions that can happen on proteins, and that they're very diverse, but mostly occur on very specific reactive side chains, lysines and arginines, that are nucleophilic and can react with these electrophiles. And today I will be focused on glycation, primarily MGO glycation right here. And many times people associate glycation with things like creme brulee or, the browning of your steak. And if you're a little more into it, you might recognize that glycation is some of the driving force behind wrinkles, as these adducts can form crosslink that crossing the extracellular matrix, the collagen, and forming the decrease in elasticity in the skin. But glycation is many times the hallmark of diabetes, and actually used for diagnosis in the form of A1C, which is the glycation of the hemoglobin in the blood. Now, due to the half-life of hemoglobin, actually detecting the level of glycation of this protein can tell you about the fluctuations or the increase in sugar levels in the blood in the past few months. But these are all very, very exciting glycation events. But how do histones play a part into that? And the reason why we became interested in histone is because they are highly abundant proteins that have very long half-lives and have highly reactive accessible tails that are enriched with lysines and arginines. So when I came and introduced this project to my postdoc, Ching Fei, we started looking at glucose. Now, glucose exists in an equilibrium with the open chain form and is a reducing sugar, and can react with lysines and arginines on proteins and create these shift-based adduct that is a little less stable and rearranges to an amidori product. However, glucose can also be metabolized into other many downstream metabolites, but one of the more interesting ones is called methylglyoxal, or MGO, that really forms during anaerobic glycolysis conditions and reacts very quickly with arginines faster than lysines, but also with lysines to form similar adducts. Now, both of these types of adducts can rearrange to what we call advanced glycation end products. And those include also cross-linking between proteins and other macromolecules. Now, we call these shift-based and these early intermediate adducts, are more reversible, and these later advanced glycation end products are really terminal products that are really almost irreversible. And even though those are the early intermediates are relatively stable, there are proteins in the cell that evolve to remove them and address their damage. One of those is DJ1, and I'll be talking about it today, is a deglycase. And the other one is FN3K, that is a kinase that actually phosphorylates this hydroxyl and shifts to equilibrium. So it's not a deglycase per se, but it does participate in regulating these adducts. And due to the lack of time, I'll really be focusing on one story where we looked at MGO glycation and DJ1 and its removal by DJ1. And the reason why we are particularly interested in it is because MGO is very abundant in cancer cells. So because cancer cells use anaerobic glycolysis, they generate lots of MGO, and normal cells have very low levels of MGO. So that was a very interesting shift in equilibrium for us here at Sloan Kettering. We do have another story on cyclic reducing sugar and ribose just published in JACS, and some hints about FN3K in it. So if you're interested, you're welcome to read that. But I unfortunately will not have time to talk about it today. Okay, so Qingfei, the first experiment Qingfei, or Grumpy Cat as we call him in the lab, did is a very important experiment, and I always show it first because many people ask, again, why histones? I gave you all the rationale, but people want to see that this rationale really holds place. So what Qingfei did is he actually took cells and he treated them with methylglyoxal for a short time. Then he chased it with media-free, MGO-free media, and fractionated the cells and analyzed the components, the soluble fraction, so that's all the proteins in the cell except for chromatin, and then the chromatin fraction. And what we found was really interesting. What you see here is the anti-MGO signal in red, and this is the soluble fraction. And in the soluble fraction, you can see that the MGO signal is generated, but is chased off very quickly within 12 to 24 hours. This is primarily due to protein turnover. But what happens with histones is that histones remember. So even after 12 and 24 hours, which include a cell division in 293s, these marks still retained and are transformed or transmitted from mother to daughter cell. And this was really exciting to see that histones are indeed able to undergo glycation, or that there are targets for glycation in cells. But we really wanted to see whether there is a physiologically relevant state where these glycation accumulate without the addition of external MGO. So that means that there is enough MGO to accumulate on histones and might drive any phenotype. And being here at Sloan Kettering, we're very fortunate to have access to breast cancer patients that have high metabolic state. And we teamed up with Sarat Chandra-Lapati here, an oncologist at Sloan Kettering, that gave us non-tumor and tumor samples from 5 different breast cancer patients. And we extracted the histones, and we're very excited to see, or interested to see, that indeed all tumor samples contain a high level of histone glycation compared to their adjacent non-tumor samples. Now, this gave us the physiologically relevant aspect of histone MGO glycation, but we still really wanted to mimic this in vitro or ex vivo so we can study it better. And for that, we took a library of breast cancer cell lines, here shown on the right, compared to a standard 293 cells, and we treated them with increasing amount of MGO. And similar to what we saw with the 293s, that there is an accumulation of glycation in the histones, there is a concentration-dependent accumulation in the breast cancer cells. But what was interesting to see is that they accumulate MGO glycation much, much faster. And in fact, they also have basal glycation. So that means that even in the absence of MGO in the media, there's enough MGO generated in the cells themselves to accumulate this signal on histones. And again, this is a Western blot that is not a very sensitive assay. And this led us to sort of believe in this mimicking system where cells under metabolic stress can mimic what happens in the tumor. But the question is, can histones really, can we analyze the biochemistry or the dynamics of this glycation in vitro? And we sort of went back to basic. We took our free core histones and we incubated them with MGO, and it was very interesting to see that despite all the histones having very similar sizes and composition, that H3 and H4 are actually much more prevalent to undergoing this MGO glycation. And in fact, when we assemble these core histones into nucleosomes, we see a similar trend. So even when they are assembled into their fundamental nucleosomal unit that is physiologically relevant, where some interactions are hidden, there's still H3 and H4 are the prime targets. But what we could see here is that there's also the formation of these AGEs, or advanced glycation end products, that include these cross-linking events. And what we asked ourselves, if there is cross-linking events, this could have detrimental implications in epigenetics or in chromatin in general, because as I told you sort of in the first slide, the capacity of chromatin to undergo dynamic compaction and decompaction is at the heart of its regulation. So in order to analyze whether cross-linking events can happen and ask if DNA is involved in these cross-linking, we went back to our nucleosomal substrate, reconstituted nucleosome, and we treated it with MGO, and we asked the question whether we get less PCR signal when we use this as a template for PCR, because the DNA polymerase in the PCR reaction is actually sensitive to DNA damage, to adducts. And indeed, that's what we saw, is if we treat our nucleosomes with MGO, we get less and less product. So that suggested that the DNA undergoes modifications. And in fact, there was a paper a few years ago in Science that shows that guanine can undergo glycation too. And this sort of led us on a route that I'll get back to in a few slides. So if guanine gets glycated even in a chromatinized DNA, and we know the histones get glycated, are these cross-linking events cross-linking histone and DNA? And to address this question, Ching-Fei used a very, I would say, elegant, and I can say elegant because it was totally his idea. His idea was that nucleosomes are held completely by electrostatic interactions between histones and the DNA, the positive charge on the histones and the negative charge on the DNA. So that means if we take nucleosomes and we treat them with high salt, we completely disassemble them. However, if some of these glycation leads to cross-linking events, a high salt treatment should not disassemble the nucleosomes. And that's exactly what he sees on this native gel. He sees the DNA, this is the H3, and this is the MGO signal. As he treats these nucleosomes with increasing amount of MGO, they undergo MGO glycation, and they don't disassemble when we reach a high enough concentration. And so this was really interesting finding to see that DNA and histones can undergo cross-linking, but this was on a mononucleosome level, and we were very interested to see whether this will affect the chromatin fiber. And I will not get into all the essays we've done. I'm only going to mention a couple because this was a really interesting sort of biochemical rabbit hole for us to try to understand what happens to a chromatin fiber when it's undergoing glycation in vitro. And the first essay we did was optical tweezers. We collaborated with Shisha and Liu at Rockefeller University. And the idea here is you take a chromatin fiber, you trap it on between two beads, and then you pull one bead away and you track the ejection of the octamers from the fiber. And this is a single molecule trace, and you can see here that in blue is the untreated and in red is the treated. Every one of these jumps is an octamer being ejected from the chromatin fiber. And you can see that when you treat these fibers with MGO, you need to exert a lot more force to eject the same, to reach the same distance, which means to eject the same number of octamers. And this is, again, just a single molecule trace, and this is the average of about 300 different single molecules, showing a difference in force that needs to be exerted. And that meant to us that there's indeed some compaction or some cross-linking happening here. Now, again, we did several different biochemical assays, but I just want to show you that if you, even though use a low-resolution assays such as MNAs just digest the DNA between the nucleosomes, you can start introducing different levels of glycation. So in this case, we treated these arrays with either low concentration or short time of MGO, and what we found is actually the fiber undergoes decompaction in response to a low concentration treatment. But the high concentration treatment that was mimicked by these optical tweezers is indeed inducing array compaction. And this led us to this biphasic model where MGO glycation on histones, when it happens at low concentration, so it's not very abundant, it actually blocks the positive charge on lysines and arginines, causing fiber relaxation because it competes with the electrostatic interactions with the DNA. But once you reach a high concentration, that means that they're in high proximity, they can form these cross-linking. Now you get fiber compaction. And this fiber compaction as well as decompaction can affect the accessibility of the DNA and the transcription in the cell, which led us to move again into cells and ask the question, does the structure of chromatin change in response to MGO glycation? And for that, we use the ATAC-seq. ATAC-seq just measures, similar to MNAs, it digests the DNA between nucleosomes and shows the accessibility of the DNA. And after an overnight treatment, what we found is that MGO induces a fiber compaction because the DNA is less accessible. And especially this is true in transcription start site. So this is the MGO treated, and this is the accessibility going down in transcription start site. So despite this MGO being a sugar metabolite, it actually induces a decrease or repression in many genes in the cell. Now, this was it was an overnight treatment, but that suggested that a similar effect to what we saw with the optical tweezers and with the long-term treatment with MGO on chromatin fiber, it actually leads to chromatin compaction, transcription repression. Now, because these adducts occur on lysines and arginines, we immediately asked the question, do they affect or compete with enzymatic modifications? And we didn't do a full analysis. We did a sample of several of these histone modifications that we're interested in, that I kind of alluded to in my first slides, that are mainly associated with transcription activation. This is, for example, A3K4 and acetylysine, but also other transcriptional events such as R8 methylation. And what we noticed is that there is for the most part, a large decrease in these modifications. We suggested that there is some sort of a competition between glycation that goes up and these other enzymatic modifications going down. But to dive just slightly deeper into this, we decided to treat the cells with, to manipulate the acylation level in the cell and see whether we can detect this competition. So we took cells, we pre-treated them with HAT inhibitor, or histone acetyltransferase inhibitors. And indeed, when you treat cells with HAT inhibitors, you get a decrease in acylation. And these cells are much more sensitive to glycation. And in fact, they start getting these basal glycation similar to breast cancer cells. And if you treat these cells with HDAC inhibitors, we stabilize the acylation and we decrease the glycation by a lot. Now, this was, again, another support for this hypothesis of the competition between acylation and glycation, but also led us to maybe a more hand-wavy hypothesis that acylation, that many times is not known to have a specific function, could actually, because its dynamics, be protecting lysines in the cell or in chromatin specifically. So that is an interesting concept, that very robust acylation is protecting the nucleophilic capabilities of these lysines. During the review process of our paper, a beautiful PNAS paper by Jim Galligan was published that taken a more metabolomics and mass spec approach. And what they found is that indeed these MGO glycation occur, and they found very similar trends where H2A, like in our hands, undergoes very little glycation, but H3 and H4 undergo massive glycation in similar sites that we have identified. So that was very, very exciting. But what we wanted to see after we saw the detection of these specific sites by mass spec is ask, can we really map all these modifications? Because glycation, and that's one of its complications, rearranges. And it's very difficult to track it by mass spec or the entire library of adducts. So what we decided to do, we decided to take a chemical biology approach and develop an AlchemGO probe that I'll just quickly mention was published recently in JOC, and mimics very nicely MGO and actually shows very similar pattern as well as cellular penetration and reactivity. So that was really exciting, and we're now using it to map the modification on histone as well as other proteins and DNA. So hopefully I convince you by now that histone glycation occurs and accumulates in metabolic stress cells. But the question is, how do cells deal with such damage? Because this has been associated with disease. And I mentioned before that there was a paper published in Science where guanine was detected to be glycated. But what I didn't tell you, because I didn't want to steal my own thunder, is that DJ1, which is an enzyme that is very abundant in many, or almost all the cells in our body, was shown to be able to deglycate these guanine glycation adducts. And since it's found in the nucleus and it deglycates DNA, immediately we ask, can it deglycate our histones? And taking a very in vitro biochemical approach, we purify DJ1 as well as its catalytically inactive mutant. And we show that indeed DJ1 can remove glycation from H3, but not its catalytically inactive mutant. And similar in cells, DJ1 can rescue glycation as well as some of these epigenetic damage that we have identified, but not the catalytically dead DJ1. And in fact, DJ1 exists in all of our cells. So if you knock down DJ1 in 293s, you get basal glycation even in the absence of MGO, again, going back to this mimic of the breast cancer cells that start accumulating MGO glycation. And there was a lot more work involved in DJ1, and it's a really interesting protein. But Qingfei was really interested in seeing whether this was the only deglycase, or maybe there are other proteins that might be competing or removing these glycation adducts. So he turned his attention to PAD4, that PAD4 actually facilitates the arginine citrullination. The original thought was that citrulline will be much less active or less sensitive to glycation than arginine, so it will probably be competing with glycation. And what Qingfei did to test this is he took nucleosomes, he treated them with PAD4, and then asked, are these more resistant to MGO glycation? And indeed, that was a very easy experiment. Indeed, if you citrullate these nucleosomes, they are less sensitive to glycation. But the more interesting question was, does it work the other way around? So if we treat nucleosomes with MGO, are these more resistant to PAD4 or DJ1? And this experiment was really, really exciting because we did either co-incubation or short, which is these early intermediate adducts, or long, so these AGEs. And we found that while DJ1 is able to remove these short-lived or these early intermediates, it really has a hard time tackling these AGEs. But PAD4 is able to not only remove these short, but also long-term adducts on the expense of citrulline. So basically, and of course together they remove almost all of these adducts in vitro. But what was interesting and what we found, and there's a lot more data in the paper, what we show is that PAD4 is actually a rewriter. So it not only removes MGO glycation, but on the same site it adds or it deaminates or citrullinates the same site. And we show that also in cells. So if we treat cells with MGO and then transfect them with PAD4, it is able to reverse some of these metabolic or some of these MGO adducts in cells. Okay, so I'm going to use the last couple of minutes to talk about cancer because DJ1 is overexpressed in multiple cancers, and it's actually the hallmark of breast cancer. That's what pathologists use to diagnose breast cancer. But it's not really known what is the mechanism of its oncogenic activity. And seeing its role in protecting chromatin, we hypothesize that cells under metabolic stress actually need DJ1 in order to maintain their chromatin intact or well enough to proliferate. And that's exactly what we saw when we took 293 T cells and we overexpressed DJ1, and we treated them with increasing amount of MGO. They deal with MGO damage much, much better than normal cells. And of course, if you knock down DJ1, they do much worse. And what is interesting is we also see the same with PAD4. And if we go back to our breast cancer cell lines, indeed, they all overexpress DJ1. But also interesting, all the tumors from our patients overexpress DJ1. But even more exciting, if we go to PAD4, all these tumors also overexpress PAD4 and in fact have high levels of H3 citrullination, which again leads to this really interesting concept where DJ1 and PAD4 might participate in protecting chromatin in these breast cancers and making them addicted to it. And indeed, when we generated DJ1 knockdown xenografts and put them in mice, DJ1 knockdown tumors grow much, much slower compared to these aggressive breast cancer tumors. Now, of course, being at Sloan Kettering, we started targeting DJ1 and developed a robust new methods for the high throughput evaluation of these inhibitors, and also already have some potential hits. And we're working with the TDI here at Sloan Kettering to try to get a lead compound for targeting DJ1. Now, just to summarize what I've shown you so far is that chromatin can undergo MGO glycation damage that can be reversed by DJ1, but perhaps there is more long-term damage that cannot be reversed by DJ1, but perhaps by other enzymes. These glycation competes with other enzymatic modifications, for example, lysine and arginine methylation. But that PAD4 can actually reverse some of these early and perhaps these later adducts to citrulline. And in fact, there was a paper published that PAD4 can also compete or reverse these methylation to citrullination, which also raised the question whether citrullination is some sort of a metabolic clock for these histones, because citrulline is actually irreversible. And lastly, this is very interesting metabolic crosstalk because sugar metabolism, methionine metabolism, and calcium dependence, PAD4, is right at that intersection of chromatin damage. And to put this all in context, I've told you that histone modifications play an important role in regulating chromatin accessibility in cellular transcriptome. But I hope that I convince you today that the chemical environment and metabolites can add another chapter for this histone code that we are trying to address using new chemical and chemical biology tools. And with that, I'll stop and I'll thank my incredible lab. Unfortunately, our most updated lab picture is on Zoom. I have incredible collaborators and I have great support by various mechanisms, and we couldn't have done this without them. And this is our lab information in case you want to learn more about my lab. And I guess at this point, I'll stop sharing and I'll take questions.
Thank you, Yael. That was really fascinating, and it's always great to learn more about epigenetics and especially the influence on histones. There haven't been too many questions, so I'll just start with one. You mentioned that citrullination is irreversible. Can you talk a bit more about the influence of citrullination on cancer state? Because I wonder then, if this influences oncogenesis or other processes in the cell.
And that's a fascinating question that we're kind of, again, it's our own little rabbit hole because there's very little known about citrullination in general. There are maybe two very high impact papers, but only really two papers that looked into the effect of citrullination on chromatin. And they have mostly suggested that it causes chromatin decompaction and transcription upregulation, which is very typical of cancer cells. But it's more been associated in these papers with cellular differentiation. It's not really known exactly the mechanism of how citrullination is affecting transcription, as well as the entire mechanism of readers and erasers. No erasers are found, no readers. I have to say, I have attempted to find an eraser before and could not. I don't know if there is one. Ching-Fei is convinced there is, so he might find it in his independent career. Stay tuned. Yeah, I think it's a really, really interesting question. Even the PAD4, it was completely serendipitous. We just thought actually that citronation is going to prevent glycation. We didn't think it's going to happen the other way around. So this was a really serendipitous finding and very, very exciting for us.
Thank you, Yael. There's the first question in the chat from Kurush Ibrahimi. I hope I pronounced that correctly. Is there any evidence that these modifications initiate cancer cell formation rather than genetic mutations?
Oh, that's a great question. I don't know if he was my reviewer, but I completely agree. But I think that I would say the caveat or the holy grail in epigenetics in general is causality versus correlation, because not just glycation, but other histone modifications, it's really hard to prove that they indeed cause changes in chromatin rather than just be associated with them or be a consequence of them. And I have to say my lab has been trying to use these INTIN technologies to install site-specifically modified histones in cells to ask the question whether they themselves drive changes in chromatin and in transcription. But I think the jury's still out on that. I do think that there is an accumulated evidence that there is causality with enzymatic ones. With the non-enzymatic one, I hope me and maybe Jim out there will do that too. We think that because of the biophysical analysis that there are direct effect glycation has on chromatin and changes in transcriptome. There are also other effects that are metabolic that could contribute to that. So I would say my hunch or my preliminary data says that there is a causal role for these, but we'll have to wait and see.
Thank you, Yael. There's another question about the difference somehow between H3 and H4. If it's, if the difference is about the sequence or the motive or the structure or really just accessibility.
We've been wrecking our heads around the difference between the different histones, not just H3 and H4. I think it's a great question, but also H2A and H2B, because really some of these experiments we have done in completely unfolding condition in guanidine. So there's really no accessibility issue. I can hand wave about like local pKa's of these lysines and arginines, but the truth is I don't have an explanation because it gets even more complicated because we thought linker histones, which is the biggest histone of it's not a core histone, it's a linker histone, but of the four, it's the biggest. And it's very enriched in lysines and arginines. And we thought in cells it will be the most accessible because it's not bound by DNA. It's actually associated with the DNA. But in fact, H1 doesn't get glycated at all. Not in cells, not in vitro by itself, it just doesn't get glycated. And so that's a mystery to me. I don't know if anyone has any idea. I'd love to hear it because we've been wrecking our brains around it.
Thank you. Another quick question about MGO and the concentrations in the experiments versus in cells.
Yeah, we get this question often, and I think it's a very important one. And sometimes I forget to say it, and I apologize for that. So basically, MGO levels in normal cells is very, very low. Single-digit micromolar. But in cancer cells, it's been measured to reach up to 500. And what we use in our experiments is we use around 500 micromolar for most of them. Sometimes we go up to 1 millimolar. But what we have done a lot to sort of show that this mimic is physiologically relevant is take low concentrations for a long period of time and show that we get a similar effect as a short exposure of high concentration. So basically, we're mimicking this long-term exposure because in cells in our body, they have really long-term exposure to these sugars and byproducts. and we didn't have the patience to wait 4 or 5 weeks for these cells to accumulate. And in fact, in or ex vivo, there's a problem that cells divide, so you get dilution of these adducts if you don't constantly treat them. So we, I think, convinced ourselves, and luckily the reviewers, that a higher concentration treatment for a shorter time is a very good mimic for a long-term incubation of a lower concentration. So,
thank you, Yael. Another question from one of the speakers, Vishal. How stable is the histone assembly, and what challenges do you anticipate if you want to install a probe site specifically?
Oh, many challenges, and chromatin is a very complex substrate. But when we do these in cells, and in fact, we already have really beautiful preliminary data that this can be done in live animals, we really try to control the site of modification by controlling the histone itself and the histone variant that is being expressed with the intine. And then the second level of control is the concentration and timing of the delivery of the second half of the intine with the adduct. So I can't say it's a perfect system, but we have learned to manipulate it enough that it gives a substantial effect, but it's not non-physiological. But again, I could talk about installation of histone modification in live cells forever. In vitro, it's a lot easier and a lot more controlled, and we have, very homogeneous substrates. But in cells, I think and I think that's where these experiments are more critical. It's a little more complex, but we're underway.
Great. Then just one last question before we move on. There are quite a few lysine sites on linker histones. Do you think there are also targets of glycation?
So that's what I was trying to sort of elude my own frustration and my students' frustration, because I told her, I was like, I'm sure it gets glycated. It has to undergo glycation. Just take H1, take MGO, and you'll see glycation. There's really almost any protein in vitro undergoes glycation except for H2A. But no, there's really nothing. It's really, really hard to induce linker histone glycation, even though there are more lysines and arginine than any other histone. And I don't know. At first, we thought it might be related to its propensity to precipitate. To aggregate. But no, we have different concentrations. We have CD showing that it's folded and in solution. No explanation whatsoever to why linker histones are resistant to glycation.
Okay, I think with that, in the interest of time, we'll move on. Yael, I think there are a few more questions in the chat that you maybe can go back and write with. Great. And then let's thank Yael once again. Now would be the time to clap. Unfortunately, I would love to have this little clapping sound, so I'm going to need to work on that for the next symposium. And now let's move to the next speaker of today, which is Jeremy Baskin from Cornell University. Yes, Jeremy is a native of Montreal, Canada, and he received his PhD at UC Berkeley with Caroline Bertossi. Shortly after, he did a postdoc with Pietro De Camilli at Yale, where he developed his passion for lipid and membrane biology. Since 2015, he's leading a team of highly skilled individuals at Cornell University. The Baskin lab uses chemical biology tools to probe the cell biology of various classes of lipids with a focus on developing new molecular imaging technologies. Jeremy has also won numerous awards, and personally, I'm really a big fan of his team's research because lipids are ubiquitously present in so many cellular processes, but there's actually so little chemically we know about the details of those processes. Jeremy, again, it's great that you're here with us today, and we are very excited for your talk. The stream is yours.
Thank you so much, Ruben. I really appreciate the opportunity to share our lab's work with everybody. Okay. So I'm on slide 1 and it's presenting. And so if I don't hear any shouts from you, I assume that everything is going
Looks good.
Great. Thanks so much. All right. So hello, world. It's morning here in Ithaca, New York, and it's a beautiful fall day. And here's a picture of Cayuga Lake and part of the Cornell campus to provide the backdrop for the title slide. So today I'm going to tell you about our work on chemical biology and cell biology of lipid signaling. And hopefully you'll appreciate the mild pun in the title as we continue. So let's see. I might as well start at the beginning. Okay, so a fundamental property of eukaryotic cells is the compartmentalization of all of their components into membrane-bound organelles. And this allows, of course, specialization of biochemical functions and different reactions to occur simultaneously in different environments within the same cell. So the membranes. The membranes that make up these organelle and cell boundaries, they're not just static entities. They're not just barriers. But there's really interesting dynamic biology that occurs on them. And so how fundamental and important is this information? I recently became a father, and as I watch my daughter grow up, of course, way too quickly, and run around the room and everything, I'm constantly reminded that she is a person that was once a cell. So how does this incredible process happen of development? This is a movie from the Keller lab at the Epigenetic campus, and it shows the development of a mouse embryo. Each cell is marked in blue, and the flashes are cell division events. How does the cell decide when to divide? And some cells have to migrate. How do they decide where to go? And then how do they know when they're there? It looks random, but there's a lot of coordination. So cell division, migration, differentiation, all of these decisions have to be made inside of an organism, but at cellular levels. And it's really how the cells talk to each other through signals from cell to cell and within a single cell that are so important for orchestrating this whole process. And these signals can be small molecule metabolites, ions, peptides, proteins, and yes, hydrophobic molecules like lipids. These signaling agents play critical roles. They perform critical roles. They provide cues. They really orchestrate the whole process. And it's no wonder that we love to use musical metaphors like play and perform and orchestrate. The signaling allows the many parts to act as a single unit, a single organism, just like a well-rehearsed orchestra. But like any high-level orchestra, there's not just one signaling agent, but there are many. And how does this symphony of information, get coordinated so it sounds like Beethoven and not like Schoenberg? Okay, so let's get back to science. Lipids, which are the hydrophobic metabolites in our cells, and many of which are the major constituents of membranes, are famous for their roles as energy stores and for components of the membrane. But certain privileged lipids, some of the structures of which are on the slide here, they can serve as second messengers or signaling agents that allow cells to sense and translate the chemical and biochemical cues from the environment into changes in cellular behavior. So what exactly do I mean by signaling molecule? Okay, so in many cases, the lipid gets generated as a signal and engages specific effectors downstream of cell surface receptor activation in order to change the physiology, the gene expression, and the overall behavior of the cell. But there are only a relatively small number of signaling agents and many, many different changes in gene expression programs and cytoskeletal rearrangements and cellular behaviors that can be occurring. So how does it work that you have a small number of signals and a large number of outputs? How is the specificity achieved? And those are really, the overarching questions that motivate our work and the research in our lab. So to put it in a broader context, if you pull open an old school textbook, a lipid bilayer might be drawn like this, where both leaflets and all the lipids look identical. But a slightly better representation might at least be this, where you can recognize that through different head groups and not really indicated different lipid tails, there are different lipids. And some are more abundant, some are less abundant, some are restricted to one side of the bilayer, so they face a completely different environment. Let's say that the cell makes a particular lipid, the red signaling lipid. It's being made in a particular membrane and a particular organelle at a particular time. The kinds of questions that we're interested in is, where are the important signaling lipids being generated inside of the cell, or in which cells in a population? What are the factors inside of the cell that regulate their production? And what are the consequences of making a particular lipid at a particular place in the cell at a particular time? So those are the questions that drive all the research that goes on in our lab, and I don't have time, unfortunately, to tell you about all of it today. The types of lipids that we have focused on so far have largely fallen into two classes of signaling phospholipids. The phosphatidic acids, which are defined by having this phosphate monoester, this small phosphate monoester head group, and the phosphoinositides, which have phosphorylated phosphoinositol head groups, which are much, much larger. And these are, of course, both classes of molecules because of differences in the case of the phosphoinositides, both in the head group in the acyl tails, and for the phosphatidic acids in the acyl tails as well. And what we realized when we started out is that there was a real lack of tools to be able to answer those kinds of questions for the phosphatidic acids. And that has really motivated a lot of chemical biology tools development in that area. On the phosphoinositides, we focused more on their role in recruiting reader proteins. And unfortunately, I don't have it to affect cell signaling. And unfortunately, I don't have time to tell you about that work today. So today, I'm going to give you some snippets of our work on developing tools to be able to study and understand and reveal the cell biology and the cell signaling initiated by phosphatidic acids. So maybe I should start by telling you a little something about phosphatidic acid. Here's a structure of a phosphatidic acid that has particular acyl tail composition. The first thing I'll tell you is that cells have three different biosynthetic pathways for making phosphatidic acid. There's the de novo pathway, which derives ultimately from glycolytic intermediates through acylation, and that occurs in the endoplasmic reticulum. And that makes phosphatidic acid as an intermediate toward the synthesis of other more complex lipids, and its levels are normally kept low. But downstream of cell surface receptor activation, there are two different pathways for synthesizing phosphatidic acid acutely and in larger but transient boluses of phosphatidic acid for signaling. And it's these signaling pools of phosphatidic acid, which are thought to be made in a variety of different membranes, including the plasma membrane and several other organelle membranes, that are responsible for recruiting effector proteins and leading to all kinds of downstream effects. So again, the types of questions that we wanted to know was, how does this one molecule or this one class of molecules mediate so many different biological functions? And so to put it in the framework of the questions that I just posed, where is the phosphatidic acid generated in the cell when particular stimuli are added? How is the production of phosphatidic acid regulated? And what really are the consequences of making phosphatidic acid in one membrane versus the other? And we realized that in order to answer these questions, we needed to develop two complementary sets of tools. One set of tools for visualizing phosphatidic acid production, so we could image in the cell where it's being made, and another for perturbing it so that we could intentionally make it in one place or another and see what the consequences were. So today I'm going to tell you about our advances to address both of these types of questions. And I'll start with the visualization tools. This is work that was carried out by two fantastic graduate students in the lab, Tim Bumpus, who just defended his PhD and was one of the first students in the lab, and Dongjun Liang, who joined the project 1 year later. Okay, so let me tell you about what Tim and Dongjun did. And in order to do so, I think I need to tell you a little bit more detail about one of those biosynthetic pathways, which is the one that we focused on, which is the synthesis of phosphatidic acid by phospholipase D, or PLD enzymes. So this is a major pathway for synthesizing phosphatidic acid for signaling. These PLD enzymes get activated by all sorts of upstream signals that derive from cell surface receptor activation. And they hydrolyze the most abundant phospholipid in our cells, phosphatidylcholine, using water as the nucleophile, to make phosphatidic acid and release the choline head group. Now, it was known for quite some time in the PLD world that if you fed cells small aliphatic hydrophobic alcohols like ethanol or butanol, the PLDs would catalyze a second reaction called transphosphatidylation. So that, as an organic chemist, you look at ethanol and you look at water and you say, well, they look pretty similar in terms of their nucleophilic character and their size. So the PLD will use the ethanol or the butanol as the nucleophile to make phosphatidyl alcohol lipid products. So these lipids are non-natural because this red part is not a natural part of a lipid head group. And these lipids have different polarity than the other phospholipids in the cell. So by radiolabeling and TLC analysis, for example, when cells are fed alcohols and their PLDs are stimulated, these lipids can be measured very accurately by the new spot appearing. And this can also occur by HPLC and mass spectrometry to identify the masses of these lipids. Okay, so the miracle of this assay is that in an aqueous environment with 55 molar water, that you can add small, low concentrations of alcohols, low millimolar, high micromolar concentrations to get efficient transphosphatidylation. So it's 9:54 a.m. in Ithaca, but it's got to be happy hour somewhere. And so what I like to say here is that, like many of the scientists who study PLDs, these enzymes prefer alcohol to water. Now, the flip side is that the tragedy of doing this traditional assay is that we lose all the spatial information. This reporter lipid is made inside the cell at the membrane where the endogenous PLD is activated, but we lose that information with the bulk biochemical measure like TLC or HPLC. So when we started our work, we really wanted to apply our chemical biology know-how to adapt this assay so that we could image lipids like this inside of cells in the membranes where they're being made. And so that's how we came up with the idea for IMPACT, which stands for Imaging PLD Activity with Clickable Alcohols via Transphosphatidylation. And it's a two-step labeling, like so many other great, fantastic two-step metabolic labeling click chemistry procedures that have been developed by the chemical biology community. In the first step, we replace ethanol with a clickable alcohol so that the PLD can perform transphosphatidylation in cells to make a phosphatidyl alcohol that we can't yet see, but that we'll be able to see after a second chemical labeling step using a bioorthogonal or click chemistry reaction to append a fluorophore. And it's these fluorescent lipids that are the reporter of the PLD activity that we visualize inside of the cell. So I'm going to breeze through some of our initial published work to say that a variety of alkynyl alcohols are able to be substrates of PLDs inside of cells to enable impact tagging. For the alkynyl alcohols, we use azidofluorophores, and we require the copper-catalyzed click chemistry reaction for tagging, which requires fixed cells. So we can't see the dynamics of what happens to these lipids because we have to fix the cells before the click chemistry. To get around that problem, we decided to use azido alcohols that enable us to use cell-compatible strain-promoted cycloaddition for tagging with these cyclooctine reagents. And again, a wide variety of azido alcohols are accepted as transphosphatidylation substrates by endogenous mammalian PLDs. And this enables tagging in live cells. So what are the examples of the types of things we can see? Here's an image of a cell where we have labeling in a variety of organelles. And of course, I'll just tell you without showing you that the labeling that we see is specific to PLDs and that the fluorescent species we identify are the ones that are expected, and the concentrations we use are low enough that they don't perturb cells. What really puzzled us with all of this imaging were the localizations themselves. PLDs were thought to be activated in large part at the plasma membrane using downstream of stimuli that we were using, which included activators of protein kinase C, such as 4-bol esters. But we didn't see any plasma membrane labeling. We saw a lot of labeling in the endoplasmic reticulum, which is not a known localization for the PLDs, as well as some labeling in organelles where they were known to act. And that really got us wondering, maybe there's a problem with the time scale of our labeling. And so as an analogy for that, I'm going to put in a shameless plug for the natural beauty of the Ithaca environment, which is the town that Cornell University is situated in upstate New York. This is one of our many waterfalls. It is very large. There are 2 people under this tiny little box here, and hopefully you can see the water is flowing. But if you can't imagine that it is, it's very loud and magnificent. The analogy is, if you follow a water droplet that's near the top, and you wait a few seconds, it's at the bottom. So if you think about the lipids in our bilayers, if we make the fluorescent lipid, if we make the transphosphatidylation product and the click chemistry tagging takes a long time, then we can't get on the microscope to visualize it until it might have moved to a different membrane in the cell. It might have diffused or trafficked from one place to another. In other words, our labeling timescale is slower than the timescale of the movement of the lipids. So to get around that, we wanted to use the fastest possible click chemistry bioorthogonal reaction that there is, which is the tetrazine ligation, which is an inverse electron demand deals autoreaction between strain alkenes and tetrazines. And it has 2 key properties: the extremely fast reaction kinetics and the fact that you can design fluorophores that are not fluorescent until the click reaction happens. Now, the challenge is both of the reagents are a little big, and so it took a lot of trial and error and a lot of synthesis to find an oxo-TCO alcohol. And this one was first reported by Joe Fox's lab at Delaware. It was a non-toxic, cell-permeable substrate of the PLDs. And so this is the one that works the best in our hands. And what's really fantastic about this new protocol is that we can do the transphosphatidylation in a few minutes, and then the click chemistry reaction we can watch in real time, because we don't have to wait till it's over to rinse out excess fluorophore, because the fluorophore is not fluorescent until it gets until it does the click reaction. And that this reaction is fast enough, we can see the labeling product appear within seconds. So here's a movie which will loop. So as it eventually uploads, you'll hopefully be able to see it. It's the first movie, the first minute of the click chemistry step after transphosphatidylation. And what I hope you can appreciate is that at very early times, there's plasma membrane labeling. And then over the course of 1 minute, the label moves from the plasma membrane to the ER. And this is really telling us two things. First of all, we can quantify the internalization of the lipid from plasma membrane to ER by looking at colocalization with an ER marker over time. And that tells us the rate of the trafficking of the lipid. And if we look at the very first few frames, that tells us where the lipid was before it moved away, and that tells us where the PLDs were making the lipid. And if we look at the later frames and the kinetics of transfer, that's telling us about how cells deal with transporting unnatural fluorescent phospholipid analogs. And it's really quite fascinating. You might think that lipids made at the plasma membrane would largely get internalized through endocytosis into endosomes that eventually meet up with other types of endosomes and then make decisions about which ways to go, the classic vesicle trafficking pathways. But that's not the case. The endoplasmic reticulum, which in this cartoon is pictured in green, goes up and essentially makes little kisses, short contacts with very small distance in between, but not fusion of membranes, with all the different organelles in the cell, essentially, including the plasma membrane. And it's at those sites where non-vesicular transport of lipids by lipid transport proteins extracting and allowing the transport of lipids one at a time can occur. And it turns out that the bulk of the transport that we see, because it goes from the plasma membrane directly to the ER, is through this non-vesicular transport pathway. And it is both unexpectedly to us an assay for this type of non-vesicular transport, for which the transport proteins are still being discovered, and is really an active area in cell and membrane biology research. And it's also a reminder that, of course, lipids are components of vesicles, but the purpose of the vesicles seems to be mainly transporting the proteins around, and the bulk of lipid equilibration through the cell appears to be going through these non-vesicular pathways. And then again, the other type of application is that by using different types of stimuli of different cell surface receptors, we can look at the first few frames of the movie to tell us where the lipid is made before it traffics around the cell. And just as two examples, we activated the muscarinic M1 receptor, which is a canonical GPCR that signals through GQ pathways, which activate PLDs. And that gets activated, the PLD signaling, at the plasma membrane. And then we looked at our canonical receptor, tyrosine kinase, the platelet-derived growth factor receptor, and its signaling resulted in, even at that early time frame, largely intracellular, and that PLD activity, and that is the result of the activation of these intermediaries, these kinases and G proteins, GTPases, at different locations in the cell where they recruit and activate the PLD enzymes. And really, this activity-based imaging approach is able to tell us not where all the PLDs are, but where the active ones are making the signaling lipid. So in sum, the impact part of the talk is this is a new activity-based imaging approach where we're adding substrates of an enzyme that allow us to use click chemistry to visualize unnatural lipid reporters of PLD activity. And I talked mainly about the subcellular applications, but there are also really exciting applications for looking at regulators of PLD signaling using single cell labeling and analysis techniques. And of course, this, like the standard radio labeling techniques I told you about, is also useful to look at bulk population levels. So that's the impact side of the story. And as a transition to the second set of tools, what we realized when we were developing impact was that we could now reveal with great spatiotemporal control or precision the locations in the cell where the PLD signals were being generated. So in the last couple minutes, I'll tell you about a tool that we developed where really the goal was to ask the question, well, so what? So what if the cell makes the PA in one membrane or another? What is the downstream consequence of that? And we realized that there wasn't such a tool in order to do that. So we could flip a switch and make phosphatidic acid on demand, only in the plasma membrane, or at any other organelle in the cell. And so we decided to go and build one. And this was work of a fantastic student, Rekha Te, in the lab, who took on this challenge and decided to use a light-mediated dimerization system, an optogenetic system, the CIBN Cry2 system, in order to recruit an exogenous phospholipase D, which, as a reminder, catalyzes the hydrolysis of PC to make PA, to recruit it from the cytosol, where it has no lipid substrates available to it, to the plasma membrane, or any other membrane, where it could hydrolyze abundant phosphatidylcholine to make phosphatidic acid. So the key is that the CIBN and Cry2 are a well-validated heterodimerization pair that only dimerize in the presence of blue light and not in the dark. We can genetically, through genetic fusions, anchor one half of this pair to a membrane of interest, and the other half, through a linker, we can attach to PLD. For good reasons, Rekha decided not to use human PLD, which has membrane bonding motifs and all sorts of regulation that the cell imposes on it, as I just mentioned, but instead she chose a bacterial PLD that was well characterized to have activity, and she adapted it for use inside of mammalian cells. And so just very briefly, by having a fluorescent protein on the PLD and shining light, she can see that she's able to recruit the PLD from the cytosol to, in this case, the plasma membrane, but using a different anchor on a different membrane, we can recruit it to different types of organelles. She then used IMPACT, and this is the result of flow cytometry analysis of thousands of cells, to show that when light is shined on the cells, no matter which membrane the PLD is brought to, there is a large amount of PLD activity there, and in the absence of light, much, much less, and without catalytic activity on the PLD, much, much less as well. So she then was interested in applying this opto-PLD, the optogenetic PLD tool, to understand the biological functions of a particular pool of PA. And so of all of the different cellular effects of phosphatidic acid, which one did she decide to look at? She decided to look at the hippo pathway. The hippo signaling pathway is really important in cancer and in other cell growth and development pathways, because the hippo pathway is a brake, is a regulator of cell growth and proliferation. And the hippo pathway are these series of kinases that phosphorylate each other, and also this all-important transcription factor, YAP, to prevent it from entering the nucleus and turning on proliferation genes. There are various upstream stimuli, including, as identified very recently, phosphatidic acid, that can suppress hippo signaling and therefore encourage proliferation and growth. But these papers that identified phosphatidic acid as one of these stimuli, they left open a lot of really interesting questions, which included, what are the effects, which are the effectors of PA? Is it these proteins directly, or what upstream factors? And where in the cell is the PA being made that has this effect? And so we thought we could start to answer some of these questions with opto-PLD. So by repeating what those papers showed, looking at the localization of this transcription factor YAP, when we add PA to cells just in the media where it's able to go everywhere in the cell, it indeed causes the transcription factor to go from the cytosol to the nucleus. And then by using opto-PLD to make PA on only one organelle or another, we can see that in red, at the plasma membrane when we make PA, that has a much stronger effect on changing the localization of YAP toward the nucleus than making similar amounts of PA on other organelles. And so this establishes that this is the plasma membrane pool of PA that is the dominant, most important pool for affecting the hippo signaling. And that sets the stage for us to be able to look for plasma membrane PA effectors that are the key ones in this pathway. The last data slide I'll show you is that we decided to use directed evolution to make a panel of opto-PLDs to test how sensitive the hippo signaling pathway is, and to have a panel of enzymatic reagents that had different strengths for making different levels of PLD. And this is an example of using the single cell labeling aspect of impact in order to be able to isolate PLDs with different activities. So in yeast, we put different PLDs based on random mutagenesis into each different cell, and then did impact labeling and isolated the brightest yeast cells based on fluorescent labeling of their cell surfaces and intracellular components that were making, sorry, not cell surfaces, but intracellular components that were making the impact-derived lipids, and then using DNA extraction and sequencing to identify different mutants. So what we found were a series of point mutants of PLD, some having lower activity and some having higher activity based on the catalytic assay of PLD activity. And when we put identical amounts of those PLDs into the opto-PLD system, we found that they had differential effects on the YAP translocation to the nucleus, showing that this hippo pathway is exquisitely sensitive or tunable to the amount of phosphatidic acid that is made at the plasma membrane. So this was an exciting result that validated the use of different types of opto-PLDs for tuning levels of PA at different organelles for affecting signaling, and also reveals some interesting insights about how PA can suppress the hippo pathway. So in summary, what I've told you today is about our two complementary tools, impact and opto-PLD, for visualizing and for controlling phosphatidic acid signaling. And some of the ongoing work in the lab is to apply these to address some of the questions I didn't have a chance to get at today, including identifying regulators of PLD signaling and probing more into the biology of PLD signaling in general. So I'd like to close by thanking the amazing students and postdocs in the lab. I've highlighted the three students whose work I've talked about today. This is a picture of us arranging into a lipid particle, sort of a vesicle, sort of a micelle, kind of neither if you think about it too hard. We're also really grateful to many foundations and agencies for their support of our work. And I mainly use the second slide so that I can show you that we can assemble into a bilayer that is flat instead of curved as well. So again, thank you so much to Ruben for the wonderful opportunity to share this work with you, and I'm happy to answer questions.
Thanks a lot, Jeremy, for this wonderful presentation. It's always good to see what your lab is up to and the new tools that you have to look at the lipid metabolism. The first question comes in from Vishal Rai. What is the time resolution of the imaging technique versus the time scale of events under observation?
Right. So thanks, Vishal. That's a great question. So with the real-time impact with the oxo-TCO alcohols, we can see the click labeling results in real time, but we still have to do the metabolic step prior to that. And so we can cut down the addition of alcohol to the cell and transphosphatidylation step down to 3 minutes is the fastest we've been able to go. And you really can't get much faster than that because there is the limitation of having to pipette something in and then having it diffuse into the cell and then having to rinse it out once or twice. Even if you don't have to wait in between rinses, you have to actually aspirate in a gentle way without killing all of your cells and lifting them off the dish. So because it's a two-step labeling, the resolution is naturally going to be down to a few minutes, which is much too slow to catch diffusion, which of course happens on the second time scale. But it does appear like it has good enough resolution in order to capture the lipids at their site of generation before they traffic from one organelle to the other around the cell. And exactly what the rates of that lipid trafficking is, I think will vary depending on what type of fluorescent head group we use, because of course we're making an unnatural lipid, and what type of organelle we're putting it in. But what exactly are the identities of the transport proteins that mediate that transport? That's one of the questions that we're currently working on in the lab. But I think by getting it down from 20 or 30 minutes to 2 or 3 minutes, we can capture the localization of where the lipid's generated before it moves around. And that's a key question. That's a key advance that's important for us. One of the things I would highlight based on what Ruben gave in his introduction about how lipids, it's been a complicated area for chemical biologists to get their, to wrap their hands around, is that lipids are a lot smaller than proteins.
I think Jeremy might just have connection issues. Let's just wait for a second until he's back. Okay, I think we lost Jeremy at this point. Let's see if in the next minutes or two he can come back. If not, there he is, I think. Let's just wait for another second. Okay, I think Jeremy might have connection issues. So let's really thank Jeremy once again for his great talk. Let's hope he can come back and answer some of the questions within the chat. Jeremy, are you back? No? I thought I was hearing something. Anyway, let's thank Jeremy once again. Wonderful presentation. And let's move on to the last talk of today, which is from Vishal Rai from IISER Bhopal. Vishal received his PhD from IIT Bombay with Professor Namboothiri, working on more classical conjugate chemistry. Shortly after which he moved to the University of Toronto for a postdoctoral stay with Andrei Yudin, where I assume that this was the place where he somehow developed his passion for the bioconjugation of biomolecules. In 2011, Vishal started his independent career at IISER in Bhopal, very, very successfully, I must admit. And his team's research focuses, broadly speaking, on bioconjugate chemistry, but more specifically on chemical methods for precision engineering of proteins and peptides. Vishal is really an incredibly kind person, and I really hope that we will soon have the chance to also meet in person. Vishal, thanks a lot for taking the time today to present what your lab is up to recently. We are very much looking forward to your talk. The stream is yours.
Thank you, Ruben, for the generous words of introduction. Can you hear me clearly?
Yes, we can hear you well.
And my slides are also clearly visible?
Not yet, not yet.
Not yet, okay. Just a minute.
Now, there we go. Now we can see your screen and
okay.
You can see the first slide.
Perfect.
There we go. Thank you, Vishal.
Okay.
Yes.
Thanks a lot. So I must also thank Ruben for setting up this amazing virtual symposium series. And it's a privilege to share this e-platform with Yael and Jeremy. Now, even though Yael got to start this session, I get to conclude it. So the debate is still on that who is Ruben's favorite. So yeah, that we will be probably discussing later. Our group at IISER Bhopal is developing chemical technologies for precision engineering of proteins. And besides, we are looking into how it can impact the field of biology and medicine. Now, before I share one of the stories from our laboratory, let me tell you why such technologies are absolutely essential in a broader perspective. So the basic knowledge of reactivity and diverse aspects of selectivity with protein provide a great opportunity to contribute to the market of biophysical and biochemical tools. The fact that this segment is doing very well even with the non-selective kits, tells a lot about the potential opportunities that are out there for the selective technologies. But what really excites our research group is the potential to contribute for directed cancer chemotherapeutics by synthesis of homogeneous antibody drug conjugates or ADCs. So this provides the state-of-the-art technology, and as the name suggests, it involves a monoclonal antibody that is covalently connected to a drug or a toxin molecule through a linker. So when these ADCs are given to the patient, the job of the monoclonal antibody is to target the antigen that is overexpressed in the tumor cells but not in the normal cells. And that's the basis of targeted chemotherapeutics. After that, the overall complex of ADC and antigen gets internalized slowly, and eventually there would be a lysosomal degradation that would release the toxins, and that would lead to the apoptosis or death of the tumor cells. Now, there are a few drugs that have received the FDA approval in recent years, and they're available in the market. However, these drugs have one thing in common, that they're sold as heterogeneous mixtures with a 0 to n number of toxins installed per molecule of the monoclonal antibody. Now, it's pretty well established that this heterogeneity is detrimental to pharmacokinetics as well as batch-to-batch reproducibility. So it's not a surprise that there were several failures in case of efforts, even in the preclinical stages that came from multiple industries or pharma sector. But not much can be done to solve this problem due to the lack of enabling technologies. And in the latter part of my presentation, I'll talk about how some of our methods have addressed these challenges. Now, as we are working with such protein-based therapeutics, we also understand that they will not be accessible to a huge population across the globe simply due to the cost factors. And that is why we want to see in the long run if we can engineer a single site of a single protein inside the human body. Because if we are successful, it could provide a disruptive platform for precision therapeutics, because then small molecules can provide the specificity that has so far been possible only with the biologics, and that would be amazing. But before we can work with complex systems such as humans, tissues, or even cells, we would need knowledge and method that can enable precision engineering of isolated proteins. And once we have them, then probably would be relatively easier to move forward and translate them sequentially from one complex system to the other. Now, at this stage, an obvious follow-up question that would probably come to your mind is that do we not already have drugs that are highly specific in nature? So let's take a look at one such representative example. Kinase inhibitors are involved in critical signaling pathway in our body. Hence, it's not a surprise that they're very well investigated over the past few decades, and they have led to multiple FDA-approved drugs. In fact, more than 37 probably just in last one decade. But even the most selective protein kinase inhibitor goes for at least 30 to 100 identified targets apart from many more unidentified ones. And this is exactly the reason why these drugs have multiple off-target activities, and they do lead to side effects in the patients. So a part of our group is trying to see if we can solve such challenges in the long run. Now, the first step in this direction was to see whether it is possible to develop methods that can result in precise single-site modification of unengineered or native proteins. And in this perspective, we started by looking at proteins as a multifunctional organic compound, which is full of nucleophilic residues, as you would see here in this cartoon. So for covalent bond formation, we will typically have to add an electrophile, and now all the nucleophiles are going to compete for that electrophile, and it will have to figure out how to differentiate one type of residue from the rest. Now, once we find such a chemoselective electrophile, and let's say it is selective for a primary amine, there will be multiple copies of primary amines present in the protein. So the electrophile then will have to differentiate one primary amine from the rest, and that is what I'm going to term as site selectivity in my talk. So for precise single site modification, we would need an electrophile to exhibit both chemoselectivity and site selectivity. Now, let's say this is the most reactive and solvent accessible amine that undergoes preferential modification with an electrophile. So for the convenience, let's call such centers as reactivity hotspots. But what if we want to attach a probe at a different amine, like this one or this one? Is it even possible to have such control over selectivity? And this is what I'm going to refer to as modularity in the latter part of my talk. So with these details in the background, we asked a very basic question, that are chemical methods capable of identifying the reactivity hotspots that are present in a protein. And after our initial investigations, we learned that N-terminus alpha amine of multiple proteins, they do serve as a very good reactivity hotspot and can be targeted preferentially in presence of rest of the residues. Now, with this in hand, we now wanted to see if chemical methods can differentiate the reactivity of a residue from its multiple copies. And we were delighted to learn that not only this is possible, we can regulate chemoselectivity and site selectivity with absolute precision for modification of highly challenging residues such as lysine and histidine. Besides, we can apply these methods to structurally diverse set of proteins, install different probes of interest, and control the type of reaction or type of bonds that are involved in the bioconjugation. Now, this works really well, but it is also important to note that all these methods are still limited to targeting the reactivity hotspots that are present in the protein. And at this stage, we really wanted to see if we can go beyond the reactivity order defined by the proteins and develop modular technologies. So the potential opportunities that can be accessed with such methods was a great driving force for us to look into it. But such a technology also required us to address a combination of challenges. And to do that, we came up with a theory that involves disintegration of the challenges at hand, finding their solutions, and then integrating the knowledge together at the end. And we argued that a multi-step chemical reaction can help us in this process. And with this thought, we wanted to see if we can develop a modular technology that can precisely label more than one sites in a protein. And we were aware that if we keep targeting a single residue, we will never be able to go beyond the most reactive site. So we decided to target two residues simultaneously. And in this example, I have highlighted lysine and histidine in this protein fragment. So the idea is that if we can design a reagent with two functional groups, FK and FH, where the role of FK is to render a very rapid and reversible reaction that is chemoselective with lysine, and FH is expected to give a relatively slow, irreversible, and chemoselective reaction with histidine. So if we can have such a reagent, and when we mix it with a protein, FK is going to go and rapidly react with all the accessible lysine residues and form linchpin at multiple sites. So now the micro concentration of FH can be enhanced near a particular histidine by the way we design the spacer, and that can be utilized to regulate the site of modification. Also, in such a scenario, the desired irreversible modification would be traversing through an intramolecular pathway, keeping out the background non-selective reaction that has to go through an intermolecular pathway. So after a thorough investigation, we found that just by changing how FK and FH are positioned with respect to each other, we were able to target histidine 36 over here and histidine 81 in a completely different domain of a myoglobin without touching anything else in the protein. And the method is not limited only to myoglobin. We can take the linchpin directed modification, or LDM, to a structurally diverse set of proteins under physiological conditions. It just gives remarkable selectivity for single site modification and allows us to control the modularity. Besides, once the job of linchpin is done, we can utilize it for installation of a variety of probes of our interest. And even better, we can take it forward and we can utilize this linchpin site to capture the labeled protein on a hydrazide functionalized resin. And once we recover the unreacted protein, we can add hydroxylamine derivative of the probe of interest, and the transoximization delivers the analytically pure single site tagged protein, and we can even recover back the resin. Now, at this point, we were all set to see how LDM platform would work for the synthesis of antibody drug conjugates. But before getting into it, let's have a quick recap of the challenges that needs to be addressed with the help of a short video. So let me just get this right for you. Okay, so as we discussed, the ADCs are composed of three key components: the toxin or the drug, in this particular example, monomethyl auristatin E, the monoclonal antibody that you see over here, and finally, the third component would be the linker that's going to connect the drug with the monoclonal antibody. Now, this also leads to the first question, that how can we control the homogeneity of bioconjugation? And once we have figured that out and the ADCs are injected into the bloodstream, the bond strength that connects the toxin to the antibody also becomes a very important parameter. So this is something that needs to be regulated very well, because if the toxin is released before ADC reaches the tumor tissue, it will have off-target effects. And that's exactly why the control over chemistry and bond types, that really becomes essential. And once that is taken care of, in the next step, eventually the ADCs will reach the tumor cells. But once they reach there, they might or might not kill the tumor cells. And this is where the control over the conjugation site becomes really critical to ensure that the ADC antigen complex is formed appropriately, and then it also has an impact on how the internalization process would happen. So once that happens, eventually the complex would reach the lysosome, and the lysosomal degradation is something that's going to release the toxins, as we discussed earlier, and that's something that is responsible for killing the tumor cells, and that's exactly what you are seeing right now in the video. So to summarize, a powerful ADC platform demands control over homogeneity and drug to antibody ratio. It should offer flexibility with the chemistry and the residues of monoclonal antibody that would be involved. So one should be able to regulate the bond strength, the linker chemistry, and the site of conjugation. And this is exactly what we're trying to address with the LDM platform that I discussed in today's talk. So let me give you a couple of representative examples. So in this case, we selected trastuzumab as the monoclonal antibody, and DM1 or emtansine as the toxin. Now, this choice was also to ensure that we can do a head-to-head comparison with Kadcyla from Roche. And we were excited to know that LDM-derived ADC renders great efficacy in inhibiting the antigen or HER2-positive cells. And not only it outperforms Kadcyla in multiple trials we did, it is highly specific towards the antigen-positive cells, without showing any negative impact on the antigen negative cells. So in this case, the site of conjugation is histidine residue, but the LDM platform does provide great versatility. So for example, we can extend it conveniently to the modular and precise modification of a high frequency lysine residue, and yet synthesize very homogeneous antibody drug conjugates. So in this representative example, we attach toxin at lysine 169 and lysine 395. And again, as you would notice, the LDM-derived ADC outperforms Kettler in inhibiting the HER2 positive cells and does not really harm the antigen negative cells at all. So with this, I hope I was able to convince you that chemoselectivity, site selectivity, residue specificity, and modularity provides a very strong platform for precision engineering of proteins. And a part of my group has been translating this to live cells, and I'll be glad to discuss that part of the story at some other occasion. But eventually, we want to see how we can take this to the animal models and beyond. And all the technologies that I discussed today and a few other are accessible to the scientific community through Playable Tech. And with this, it is time to thank all my group members who have done the actual work in the laboratory. Sorry for the old photograph. Many of the guys you see in the picture are alumni. I would also like to thank all my collaborators who have been supporting our research program strongly since the very beginning. And we are very thankful to all the funding agencies for their constant support for all these years. And thank you all for your kind patience. I'll be glad to take your questions.
Thank you very much, Vishal, for the fascinating talk. I'll jumpstart the discussion with a quick question from my side. Can you talk a little bit about the yields in the reactions with your LDM platform? I think, let's say, overall in terms of antibody, but also maybe in terms of the drug coupling step. I guess that varies between the different examples, but maybe you have a bit of overall feedback about this.
So let me get it right. So you want to understand the yields of the antibody drug conjugate?
Yeah, I mean, basically the yields of the antibody drug conjugate when you look at the antibody, so overall, but also the, let's say, efficacy or yield of the drug coupling step. Because I guess that's one of the price. Yeah, okay.
Yeah, so I understand completely what you're asking. So what happens is that if you push for yields, you might compromise the homogeneity. So there is a little trade-off between these two parameters. But what happens in our technologies is that we never install the drugs in the bioconjugation step, in the first step of bioconjugation. So when we use LDM platform for that matter, we would let the conversions go till we are getting a homogeneous antibody drug conjugate and an appropriate drug to antibody ratio. And while we make these batches, we create different batches for this purpose, where we will have different drug to antibody ratios. The advantage of one of the method that I showed with protein for purification aspect and was the fact that you can capture everything that is labeled on the resin. You can recycle everything that is not reacted. So your effective yields will go up, but your actual yields will range somewhere between 10% to 50% depending on the method that is utilized. And once we have this captured pure tagged monoclonal antibody, then you use the hydroxylamine derivative of the toxin to release it. So the centrifugal spin concentration would just give you pure antibody drug conjugate without contamination from monoclonal antibody at all.
Yeah, I think 10 to 50% yield for such a system is great. So that's definitely very interesting. Another question from Kurush Ibrahimi. He asked about the homogeneity, if it can be controlled by, for example, addition of unnatural amino acids to the antibody.
For sure. In fact, there is an effort going on in that perspective as well. And the there was this remarkable work that came out from some of my colleagues in US who were trying to find out how to have multiple sites where they can install the unnatural amino acid without really compromising the activity and the binding specificities of the antibody, and they were successful, at least as per the report that came out in the publication. So yes, however, you can argue that eventually what will be the cost factors, and how are you really going to bring profit in the market if you think from the business perspective. So depending on whether the attendee comes from industry or academia, the priorities of which technology to use can change.
Okay. Now that you talk about this issue between industry and academia, can you talk a little bit about the costs, just out of curiosity?
Yeah. So normally, so this is where what we can say is that the cost evaluation at this point that we do is very difficult to say, because a lot of cost that adds up is not from synthesis part, right? The cost of the antibody obviously is one cost, and the cost of toxin, and both these things we are procuring right now from an external source. But when you look at the cost of the antibody drug, for example, if you look at the cost of Kadcyla, you would realize that it's not really that challenging to beat that in the terms of cost as well.
Okay,
so all I would say is to keep it more vague, the precision doesn't come at extra cost when you're going with the chemical technology.
Okay, so you say basically the main cost comes from the antibody expression and the drug itself, rather from the technology of combining the two.
Right.
Okay, great. Another follow-up question on the unnatural amino acids. Basically going back, I guess somehow back to the cost. So do you think that the unnatural amino acids incorporation will basically make the technology as such, with such an amino acid, so expensive that it wouldn't be something interesting for industry, because the whole process of creating a artificial antibody with an unnatural amino acid will be too complicated to be industrially relevant?
Right. So first of all, it has to meet all the parameters that you would expect a biosimilar to meet, right? And then after that, the factor comes that, okay, the antibody drug conjugate, how is that performing, right? So there are challenges, but we cannot completely rule out. At this stage, it's too early to kind of rule out the economical viability of that technology as well. So I'm hopeful that it will do well as the time progresses.
Great. Can you also talk a bit about, let's say, competitor strategies or other strategies out there in the market, how they compare to your method?
So I guess, even now, LDM is the only technology that provides modular platform for precision engineering of proteins or antibodies. So there's no other way you will be able to even regulate the site of conjugation, which lysine or which histidine do you want to connect your toxin to, how does that really impact? And if you look into some of the literature that is already out there, it does matter eventually in the pharmacokinetics that what are the sites of conjugation. So when it comes to the homogeneity, the extent of homogeneity and the modularity, I do not see any other technology as a competitor as of now. But yes, there are a lot of other investigations before we can take it to the IND level. Yeah.
Okay, great. I think if there are no further questions, I think we'll come to the end of today's session. Let's first of all thank again all of the speakers of today. I think this was really an amazing symposium, and a further highlight on how translationally focused research in chemical biology can bring the two disciplines, chemistry and biology, really closer together. Please stay with me for another minute. I'm going to announce our next upcoming chemistry symposia at the end of the event. But before I do that, a special thanks goes to all the Wiley colleagues behind the scenes, but really especially to Ananias from UJOC, who is really the main driving force behind all our virtual events at Chemist Europe. And to be honest, probably one of the best colleagues that one can wish for. I'm just going to try to share my screen now and present you the next upcoming events we have planned. One is from Chemistry Open. I hope everyone can see my slides. So that's from Chemistry Open. It's their first virtual event coming up on open source research and open data. I'm going to share the link to this symposium in a minute with talks by Matthew Todd, Romy Amaro, and Alberto Pepe from Atipon. And the next event from ChemBioChem that we have planned is a joint virtual event with ChemMedChem on chemical epigenetics. That's going to take place on December 4th. It's going to be moderated by Manfred Jung from the University of Freiburg and Oladia Vasquez from the University of Marburg. And the speakers will be Alessio Zioli, Emily Dijkhuizen, Akane Kawamura, and Julian Level. I'm also going to copy the link to the symposium into the chat in a second. And with that, I'm just going to unshare my screen now. There we go. With that, we are at the end of today's symposium. And I again would like to thank all speakers and also especially all attendees dialing in from all over the world, and also for the very lively and productive discussions today. Let's raise our glasses, marks, whatever feels right to you at the time of the day. Cheers, and see you at our next Chemist Europe event.